Hi folks! This is our third video on kinetics. I'm going to be talking about principles and postulates related to kinetics. The Hammond postulate relates to the placement and structure of the transition state relative to the starting material, product, or intermediate. Whichever species the transition state is closest in energy to is the one that it most closely resembles. So let's consider we have a starting material with this energy, and I'm drawing a vertical line as the midpoint along my reaction coordinate. If you have an exergonic reaction, the transition state is going to be closer in energy to the reactants compared to the products. We would call this an early transition state, and it would appear to the left of the midpoint along our reaction coordinate. The transition state will more closely resemble the starting material compared to the products. Instead, the transition state of an endergonic reaction is going to be closer in energy to the products rather than the reactants. This will be a later transition state, appearing to the right of the midpoint of our reaction coordinate. The transition state structure will appear more product-like than its resemblance to the starting material, because it's closer in energy to this product. Importantly, the Hammond postulate tells us about the placement of the transition state and its resemblance to the starting material or product, but it does not tell you about the barrier height. The Curtin-Hammett principle occurs when you have competing reaction pathways. These pathways could be occurring from any types of species that could be interconverting, such as isomers, different conformations, or different intermediates. The Curtin-Hammett principle states that as long as this interconversion is fast compared to the rate of product formation, the amount of each product that's formed is determined by the barrier heights rather than the difference in energy of your interconverting species. So let's consider you have two different species, A and B. These could be different conformations or different intermediates. And let's assume that they are rapidly interconverting, meaning there's a low barrier going from A to B or vice versa. Let's assume that A forms a product A, and that this reaction is slower than the conversion of A into B. And let's assume that B forms a product B, and that this product formation is slower than the interconversion between B and A. Let's assume that we have a scenario where B is lower in energy relative to A. I've drawn a low barrier between these two to suggest that they would convert back and forth quickly. One scenario is that there's a high barrier going from B to product B. I've denoted this by a large delta G double dagger going to B. And let's assume that we have a lower barrier going from A to product A. Here, this delta G double dagger value is smaller, corresponding to a lower energy barrier and a faster rate. This is a scenario that I've laid out in which the less stable species, A versus B, leads to the major product. Product A is the major product because, by the Curtin-Hammett principle, the major product arises from the lower reaction barrier, and it doesn't have to do with the energy of A or B. Now let's consider we have the same energies for A and B, and the same low barrier going between the two. But now let's consider a scenario where the barrier going from B to product B is relatively low, and the barrier going from A to product A is larger. corresponding to a larger delta G double dagger value. In this scenario, it happens to be the case that the lower energy B leads to the major product, which is product B. Product B is the major product not because B was lower in energy than A, but because there was a lower barrier going from B to product B relative to the barrier going from A to product A. So here as well, by the Curtin-Hammett principle, our product distribution, or which is the major product, is determined by the barrier heights, not the energies of our interconverting species, A and B. As an example of a Curtin-Hammett scenario, let's consider an amine methylation reaction. Here I'm treating this amine with a carbon-13 labeled iodomethane molecule. This amine methylation will proceed by an SN2 mechanism. In the product, my nitrogen would have a new bond to the carbon-13 labeled methyl group. And notice that this cationic nitrogen product is chiral if all of these groups attached to it are different. Let's consider two specific amines, which I've labeled A and B. In the case of A, the methyl group is pointing toward the right over this 3-carbon side of the molecule, and the lone pair points to the left over the 2-carbon side. In B, the placement of the methyl group and the lone pair has changed. This occurs by amine inversion, similar to the way that we saw that carbanions can invert. This amine inversion is fast, and it's been experimentally determined that the molecule A, in which the lone pair points toward the left, is less stable than the molecule in which the lone pair points toward the right. If structure A reacts with the carbon-13 labeled iodomethane, the product has the carbon-13 methyl group on the left side, where the lone pair previously was. If instead structure B reacts with the carbon-13 labeled iodomethane, after the SN2 reaction, the carbon-13 methyl group will appear on the right side instead. It turns out that the structure I've drawn on the left is the major one, while the one I've drawn on the right is the minor one. In this scenario, A is less stable than B, and there's a low barrier going between the two. Because the product arising from A is the major product, that means there's a relatively low barrier between A and product A. And because the product arising from compound B is the minor product, that means, by the Curtin-Hammett principle, that the transition state between B and product B needs to be higher in energy than the barrier for A to product A. To reiterate just one last time, our product distribution is determined by these barrier heights and not the starting energies of A or B. The principle of microscopic reversibility states that whatever pathway you would have for going from a product back to a reactant would be the same as for the reverse pathway, going from the reactant to the product in the original reaction. This means that you would go through the same intermediates and the same transition states in either direction. As an example, let's consider conversion between an alkene and an alcohol. This reaction can proceed in either direction, depending on the concentration of acid used. In the forward reaction, we would start out with the alkene, and it would react with H3O+ in a proton transfer step. The protonation would occur at the less substituted site to make the more substituted carbocation intermediate. This carbocation is a strong electrophile, and water could serve as the nucleophile in a nucleophilic attack step. This would form an intermediate where you have a new carbon-oxygen bond, and the oxygen is still cationic. Then another equivalent of water could come in and be used as a base to deprotonate this cationic intermediate. This deprotonation would form the final alcohol product. Now let's consider this reaction going in the reverse direction. In the first step of the reverse direction, the alcohol would be protonated by H3O+ to form a cationic intermediate. Notice that this cationic oxygen intermediate is the same one I encountered in the forward direction as well. Now that it's been protonated, the oxygen is a good leaving group, and it could leave in the form of water. This forms a carbocation intermediate, and notice it's the same one we formed in the forward direction. Here I'm drawing out a hydrogen adjacent to the carbocation, and this is going to be deprotonated by water. This deprotonation forms the alkene. By the principle of microscopic reversibility, the mechanism in the forward direction or the reverse direction needs to involve the same cationic intermediates. The last topic for today is kinetic versus thermodynamic control in reactions. This is important when you're considering the relative amounts of two or more products that are made in a reaction. Under kinetic control, the ratio of the products is determined by the energies of the transition states, with the lower energy transition state leading to the major product under these conditions. Reactions are typically under kinetic control when they are run at low temperatures. Under thermodynamic control, the product ratio is instead determined by the energies of the products rather than the transition states to get there. Thermodynamic control relies on the fact that the products need to be able to interconvert or be at equilibrium. Thermodynamic control will often be in place when you have reactions that are being run at higher temperatures. We'd use the term kinetic product to describe the product formed by the lower energy barrier pathway, whereas the thermodynamic product is the more stable product. Let's consider two scenarios. In the first one, let's consider we have an intermediate that could go on to form either product A or product B. Notice I've drawn product B lower in energy compared to product A. Here in purple I'm showing a barrier going from the intermediate to product B. And here I've added the green barrier going to product A. Let's consider what would be our kinetic versus our thermodynamic product. The kinetic product arises by the lower energy pathway. That's going to the left, so product A is our kinetic product. Then our thermodynamic product is simply the lower energy product, product B. So in this scenario, our kinetic product and thermodynamic product are different. Another potential scenario is that my intermediate product A and product B energies are all the same as before, but now the barrier going from the intermediate to product A is higher than the barrier going from the intermediate to product B. In this case, product B would be my kinetic product because it has the lower energy pathway going from the intermediate to that product, And then product B is lower in energy than product A, so it is also the thermodynamic product. So it is possible for one product to be both the kinetic and the thermodynamic product. If this is the case, that is going to be the major product regardless of the temperature. In summary, today's video talked about 4 different principles or postulates. I talked about the Hammond postulate and how that relates to the placement of the transition state in your reaction coordinate, depending on whether a reaction is endergonic or exergonic. I talked about the Curtin-Hammett principle, which applies when you have 2 different species interconverting quickly and different products that can arise from them. I also talked about the principle of microscopic reversibility and what that tells us about our mechanism in the forward and reverse directions. And I ended by talking about kinetic versus thermodynamic control in reactions. That's it for this video. I will see you in class.